So I like to thank Dr. Mohita for giving me this opportunity to talk about the stem cells in cornea, senescent to recent. The stem cell research in the field of ophthalmology over the past 2 decades had advanced in knowledge and treatment dramatically. So it was initially in 1944, the presence of stem cells were indicated, and then in 1971, the pigments at the limbus, they were proposed to be stem cells, and in 1991 Tseng, they have proved that the finding of the stem cells present in the pellicid of Vogt in rabbits. So what are these stem cells? They are specialized, undifferentiated, self-renewing cells which are responsible for the corneal epithelial renewal and regeneration. They act as a barrier in preventing conjunctival epithelium from growing onto the cornea, and thus they help to maintain the normal healthy ocular surface. So when there is significant damage to the stem cell, what happens? You are seeing in these pictures. They are showing conjunctivization, conjunctival overgrowth, chronic keratitis, scarring and calcification. So what are the different types of limbal stem cell deficiency? It is primary as well as the secondary. Primary limbal stem cell deficiency can be congenital and acquired, and the secondary one, which is commonest, that is always acquired one, and the cause related to the external factors that destroy the limbal stem cells. And site of pathophysiology in secondary always lies in limbal cells. According to the extent of involvement, it can be sectoral or diffuse. Diffuse means total 360-degree loss of the limbal stem cells. So diagnosis can be done clinically or by excisional corneal biopsies or by the impression cytology. In impression cytology, one can see clearly the conjunctivization of the cornea by the presence of goblet cells over the cornea. The management is not that easy. It is different in unilateral and bilateral cases. Unilateral cases further can be divided into partial and total. Partial unilateral limbal stem cell loss can be treated with sectoral, sequential, conjunctival epitheliectomy, amniotic membrane transplantation and conjunctival limbal autograft. And total, we require conjunctival limbal autograft, cultivated conjunctival limbal autograft and simple limbal epithelial transplant. And bilateral cases, they required keratolimbal, living related or cultivated limbal transplantation. So if we see one by one, conjunctival limbal autograft, you take the healthy tissue from the healthy eye and then transplant into the affected eye. So in case where there is no healthy tissue in the other eye, one can use the cadaveric donor, and similar way we can transplant the healthy stem cells into unhealthy eye or affected eye. There is another thing where we don't require the larger amount of the stem cells, and we can take the small piece of the stem cell and cultured or cultured in the lab, and then this cultured amniotic membrane can be transplanted over the affected eye. But this requires very expensive lab setup. So these are the drawbacks in limbal stem cell transplant. If we take from the other eye, there may be loss of limbal stem cells in the donor eye. In cadaveric, we require immunosuppressant, and in lab setup, we require very expensive lab setups. So we require something simple technique which can be done at the remote places, this can, for the uniocular cases, we have simple limbal epithelial transplant, which was innovated by Dr. Sangwan in 2012, and this is single stage, affordable, and minimal donor tissue is required. And the beauty of this technique is it is repeatable, and this can be done in very resource limited settings. This is very simple procedure, as I have small clipping. So in case of 360-degree limbal stem cell transplantation, there is fibrovascular pannus or the fibrosis over the cornea. So you just remove this, clear the cornea till you get the clear cornea. And it is very important to remove this fibrosis not only from the cornea but beneath the conjunctiva, so you get a very healthy ocular tissue. Ocular surface, and then after removing this, you just put the glue over it and then place the amniotic membrane and stick it, and you can tuck the amniotic membrane beneath the conjunctiva so that further proliferation of the fibrotic tissue can be prevented. And then from the healthy eye, small strip of the stem cells is taken and it is cut into small pieces, and then it is glued over the amniotic membrane, which is placed over the clear cornea. So as the name suggests, it is very simple technique and it gives the very good results as shown in these figures. So sometimes you may have some exotropia in the cases like in this boy, he came very late, so he requires amblyopia therapy. So if we see the future of the stem cells, so it can be used in as a corneal stromal stem cells in different approaches. One can inject stem cell alone intrastromally or along with the biodegradable scaffold or non-biodegradable scaffold, or it can be used with decellularized corneal stromal scaffold. So scientists are working to research and we may get something new in future. Thank you.
Thank you, Dr. Shreya. Now